Okay, let's look at our components at an even more microscopic level, a subcellular level, where we definitely see the terminal cisternae and the sodium, or excuse me, the calcium that would be being released from there into the actual myofibril area, which has this myofilament. This is my actin filament. Or thin filament, which the big blue beads here are actually going to be considered to be the actin proteins. The thread-like protein is the tropomyosin. The protein that is attached to the bead-like protein attached to the thread, this is the troponin. And notice that the little black knobs here are the binding sites, and they are presently being covered by the troponin, or excuse me, by the tropomyosin. So here is my thick filament, which is made up of many different myosin proteins with their cross bridges. All right, now again, the terminal cisternae was part of the triad. Our T-tubule, which stands for transverse tubule, would run in the middle of two terminal cisternae to form the triad. The T-tubule is what we would send the action potential or the nerve impulse down. So when we came to the terminal cisternae, there are voltage-gated channels that if we change the voltage by sending the signal, then we'll open up the doors, allow the calcium to come out. Well, the calcium is going to come out and attach to the troponin, which is going to shift the tropomyosin threads off of the binding sites and make them available for the cross bridges to be able to come up and attach to, and then do a pivot motion that actually pulls, because that's what we're doing. Our thick filament is pulling our thin filament, and that is contraction. When the thick filament releases and this goes back, then that's a relaxation or even a stretch. So again, our components of our filaments, our thin filament is made up of primarily actin proteins, the blue beads here, the tropomyosin threads, the troponin that actually has a bonding site that will bond with calcium once it's released. But if the calcium's not there, then this tropomyosin thread is bound to one side to the troponin is bound to one side to the tropomyosin thread, the other side can bind to calcium. So as long as there's no calcium, then the troponin shifts the tropomyosin thread over the black knobs, the binding sites, and the cross bridges don't have the ability to grab and pull. All right, so the other thing that we've got here is ATP, which is used to disconnect the cross bridge from the binding site, but then it hydrolyzes into inorganic phosphate and ADP. In other words, we phosphorylate that, we break off that N-phosphate group and transfer the energy into this cross bridge. So now it's ready to spring forward again and grab a binding site. And of course, we're going to do this. This is a two-dimensional representation of an actual three-dimensional event. Okay, let's watch this event. Hopefully you will have watched the videos previous to my overview of them, but let's watch this event.
On the following pages. To see the entire process of cross-bridge cycling, click the terminal cisternae.
Here's our action potential, calcium released. Okay, now again, looking back at this and breaking it down even a little bit, just a little bit further. So what happened? We had to release send the action potential, cause the calcium to be released. We actually see that once the calcium is released from the terminal cisternae, and again, that's because there's voltage-gated channels here that once we send action potential triggers them to open up, we release the calcium. Calcium's going to bind to troponin. Okay. Once it binds to troponin, it shifts the threads off of the binding sites. Now our energized cross bridge has the ability to come up and grab the binding site. Okay, and actually pivot or pull this. Now notice now our next ATP molecule has a binding site on the cross bridge, which will literally cause it to disconnect from the binding site. And then once we split that last phosphate off, or we phosphorylate this to release the energy, well, the energy cocks us back where it's ready to spring forward again and grab that again. Now, in this situation, we literally have the calcium that was released being pumped back into the terminal cisternae. If you remember, active transport often is used for pumping things against their concentration gradient. Because even though we release calcium from here to this area to bind to the troponin, there's always more calcium inside that wasn't released until we send the next action potential. So again, this is going to, we're going to use ATP not only to disconnect this and energize it, but we're going to use ATP to pump the calcium back into the terminal cisternae. But when we do that, notice how the threads come back and cover the binding sites again. Okay, so let's look at what would happen when several of these actually grab this, because it's never just one. It's almost like a tug of war where you've got several people pulling on the rope. Okay, we see a representation of the functional unit of muscle contraction, the sarcomere, which runs from Z line to Z line. Again, the Z lines are protein discs that have our thin filaments coming off of them. Our thin filaments have our actin molecules with the binding sites, has the tropomyosin threads that cover it, and has the troponin, which is connected to the tropomyosin threads that also binds with calcium once it's released. Now, we also have our thick filament here in the middle, which is primarily made up of myosin proteins with the cross bridges. We have our dark bands, the dark area, which is the length of the thick filaments. We have the I bands, which is everything outside of the thick filaments. We have the H zone here, which is the distance between the thin filaments. And again, right now he's at a relaxed position, so there's space in the H zone. When he contracts, this space would disappear as each of these came in as they were being pulled. We would also have an M line that would be anchoring this area, even though it's not shown in the picture.
Click a thin filament to start the contraction.
Okay, notice how now our H zone has disappeared because we're contracted. Notice it's contracted. The sarcomeres within the muscle cells, within the muscles, within the fascicle, and then all those fascicles in the muscle. So we're down here to a subcellular level, but this is actually where contraction's taking place. Notice how my H zone, again, has disappeared. And, all right. Now my H zone has reappeared because now I'm relaxed. And of course, if we stretch that muscle, that may even actually, be a little bit wider. Okay, what does ATP do for us? Well, it actually energizes the myosin bridge so it can spring up and grab the binding site. It's actually used to disconnect that cross bridge once it has pulled the binding site. And then finally, it's used to transport, because it's active transport, the calcium from the troponin back into the actual terminal cisternae. Now here we see again the membrane, because this is a membrane-bound organelle, a phospholipid bilayer with a protein embedded in it. This specific protein being one of the doorways that we're trying to talk about right now, and this actually being a doorway that has to be opened by a power source of ATP, but this being a doorway that is a pump, so I can pump the calcium back into the terminal cisternae after we've used it in the contraction.
Transport.
Okay, notice how our ATP is being hydrolyzed, phosphorylating that, transferring the energy so we can literally pump it. Okay, now let's combine two major subjects we've talked about, which was cellular respiration, which we remember the whole glycolysis and the transition reaction, the Krebs cycle, and oxidative phosphorylation, which we refer to as happening in the electron transport chain. All that stuff that was part of actually test 2. Let's combine it with what's going on now, because again, we learn stuff so we can use it, not just so we can memorize it for a test and not have any idea why it's there. But let's combine what was going on with cellular respiration, which can occur in almost every cell, whether it is only glycolysis or whether it's all 4 steps. But let's talk about it as it applies to muscle cells, because they are the main place that we're going to use the oxygen that we breathe in. All right, so if you will remember, we said that glycolysis, splitting the glucose from a 6-carbon down to the 2 3-carbon products. Remember we said that we had to start out with 2 ATP. We got 4, but we had to pay back the 2, so we got a total of 2 ATP. Well, where did those 2 ATP come from? Well, one of the main sources of generating those first ATP is the fact that we keep a product called creatine phosphate. So notice adenosine triphosphate, creatine phosphate. So what we actually do is we borrow some phosphates from the creatine phosphate there, and then we turn ADP into ATP by using that creatine phosphate. Therefore, we have our first 2 ATP that now can cycle this program of glycolysis, going from the 6-carbon to the 2 3-carbon byproducts. Well, we only have limited amount of creatine phosphate, so it's used up rather quickly, but what it does, it starts this process going. I love this picture because it does show ATP, at the chemical level. And here's our adenine and our ribose, so that's the adenosine part. Here's our 3 phosphate groups right here. And even though they're actually showing the high energy bonds, they're showing it by showing the covalent bonds between the phosphate. Remember, in reality, that energy is actually held between the oxygens, the negative charges on the oxygens. So if I can break off that and phosphorylate something, I literally release that energy to do the work of whatever the cell needs. So in this picture, what we see is the hydrolysis of the creatine phosphate means that we split the phosphates off and added them onto some ADP to form the ATP. And what that did, that actually got the glycolysis product started, because that gave us the 2 initial ATP to use, and then we made 4 ATP, paid back the 2, and then of course glycolysis either leads to our pyruvic acid either going through the transition reaction into the mitochondria, or turning into lactic acid outside of the mitochondria. Okay, that once we're inside the mitochondria, we know the Krebs cycle takes place. We still get a little bit of ATP from that. 2 ATP per turn of the Krebs cycle, or 1 per turn for a total of 2 there. And then of course oxidative phosphorylation is where we get our big payoff. Of course, one of the components of being able to do this process and get ATP is the fact that we have to have glucose. So where do we get glucose? Well, we either get it from our blood, because it's going to go through our facilitative doorways from our blood into our cells, or we get it from a stored amount of glucose. We call that stored glucose, those chains of glucose molecules, we call it glycogen. And as we said before, where do we store the most glycogen in our bodies? In our liver and in our muscles. So again, here we see our glycogen reserves, our chains with a lot of side chains of glucose molecules, or get it directly from the blood. Now, which one of these we use primarily is actually dependent on whether we're looking at red or white muscle fibers, which we will be talking about in a few minutes. This picture gives us a great summary. Glucose is coming in, or it's already in there being stored as glycogen, but we have to break it off to use it, where our creatine phosphate is hydrolyzed. In other words, we break off the N-phosphate to have the ATP that's available to go through the rest of these steps.